Upon hearing the term weapons, the human mind typically conjures images of guns, grenades, ammunition, and the like. However, within the realm of biology lies a category of more lethal and detrimental weapons known as bioweapons. These weapons fall under a subset of a larger class of weapons commonly referred to as unconventional weapons or weapons of mass destruction. Not only are they associated with significant mortality and morbidity rates, but their deployment also instills public fear and panic, ultimately leading to severe economic and social destabilization. Due to this, they are among the highly regulated weapons by most governments and research centers, such as the Center for National Security and Arms Control. So what exactly are bioweapons? What is their history, and what harm can they cause? Stay tuned. Bioweapons, or simply biological weapons, encompass pathogenic microorganisms such as bacteria, fungi, or viruses, along with the respective products they produce, such as spores and toxins. It is essential to understand that during an attack, a microorganism can be used directly as a weapon, or the toxins they produce can be extracted and weaponized independently. The affordability, accessibility, ease of dispensing, and the ability to rapidly multiply inside humans, animals, or plants make these pathogens exceptionally deadly. Therefore, intentionally producing and releasing these pathogens, their toxic products, or infected material with the aim of causing death to humans, animals, or plants is commonly referred to as germ warfare, biological warfare, or bioterrorism. Surprisingly, the origin of these kinds of attacks extends back for centuries. During historical wars, some armies opted to use contagious and infectious disease-causing agents as weapons to confront and constrain their opponents. One notable incident took place in the year 1346, when the Tartars catapulted plague-infected bodies into Kaffa, Crimea, in an attempt to break a 3-year siege. Another incident occurred during the 18th century when the British forces employed smallpox-contaminated blankets to spread infection among North American Indians. Similarly, between the 1930s and 1940s, the Japanese released fleas infected with the plague in Chinese cities, leading to hundreds of thousands of deaths. Also during those times, offensive and defensive biological research programs were warmly embraced by several countries, including the United States, Britain, the former Soviet Union, and Canada. Today, some diseases and microorganisms have been classified as bioweapons or potential bioweapons. So what are they? First on this list is anthrax, a deadly disease affecting livestock, caused by the bacterium Bacillus anthracis. This disease is also classified as a zoonotic infection, meaning it can be transferred from animals to humans. Humans can acquire it through contact or inhalation of the causative agent, particularly when in contact with infected animals or contaminated animal products. In humans, this infection can manifest in inhalational, oropharyngeal, cutaneous, or gastrointestinal forms, each of which carries the potential for fatal systemic anthrax. The infectious dose estimate of anthrax pathogen is between 8,000 to 50,000 spores. According to the North Atlantic Treaty Organization classification, the anthrax pathogen is ranked among the top 4 agents with the potential to be used as a biological weapon. This particular pathogen possesses characteristics ideal for bioweapon development. For instance, it can produce highly survivable spores suitable for use in explosives. Moreover, it remains stable in various environments, and the process required for its mass production is relatively straightforward. What makes it deadly is that, once inhaled, its untreated form leads to high mortality rates, and its advanced form becomes extremely difficult to treat. Although non-contagious, anthrax can function as both a disease and a weapon. Fun fact about it is that it's not a new disease. Its reference can be found in ancient writings and religious texts, with some authors speculating and interpreting it as the 5th and 6th plague that hit Egypt during the reign of Pharaoh. Also, this particular disease was officially classified as anthrax in 1769 by Jean Fournier, who highlighted the link between people working with animal wool or hair and the high incidence of anthrax cases. Most of these cases were observed after the introduction of mohair from Asia and alpaca into textile industries in the 1800s. This was evidenced by the increased number of deaths among workers closely involved with mohair products. The use of anthrax as a weapon cannot be underestimated in historical wars. Interestingly, Adolf Hitler was targeted for assassination using an anthrax-contaminated fountain pen, a plan that would have ultimately contaminated his clothes. Fortunately, this plan did not come to fruition. This plan did not. Not to mention, during World War I, the Germans used needles contaminated with anthrax bacterium to inject horses, cattle, and sheep destined for the battlefront, sourced from various countries, including the United States. This was done before boarding the animals onto the ships. Also, just before World War II, a unit number 731 was constructed by the Japanese government to research, develop, and manufacture offensive biological weapons, including anthrax pathogen. Even though this facility was taken over by the Allied forces towards the end of that particular war, it had the potential to produce 1,400 pounds of anthrax-causative agent yearly. Its scientists and engineers employed various tricks, designing a fragmentation bomb capable of dispersing anthrax spores and even creating anthrax-contaminated chocolates. Another incident occurred in January 1917 when a Swedish spy named Baron Otto Karl von Rosen was detained for possessing anthrax as a bioweapon. Von Rosen had intended to distribute anthrax-laced sugar cubes to the horses he encountered while utilizing the government's permit to transport horses across the nation in support of the Allies on the Russian front. As the years passed, Great Britain initiated a research program in the late 1940s to explore the dispersion of bacterial aerosols. This research heavily focused on toxins from two bacteria, Bacillus anthracis and Clostridium botulinum. Field tests were conducted on Gruinard Island off the coast of Scotland, exposing a flock of sheep to anthrax spores. The objectives of these tests were to establish the optimal bacterial spore concentration that needed to be suspended in aerosols released from bomblets and to determine the extent of downwind cloud dispersion. The results of this trial test were shared 3 years later with leaders in the United States and Canada. This led to the establishment of a collaborative effort, with the British designing the weapons, the Americans constructing them, and the Canadians carrying out the testing. Interestingly, by the 1960s, the United States had accumulated 3 deadly biological agents, including anthrax. Despite this, anthrax was never utilized as a biological weapon, and the offensive program came to an end in 1969, in line with the US government's aggressive efforts to outlaw such weapons. Notably, in the year 1941, Prime Minister Winston Churchill approved a project named Operation Vegetarian, allowing scientists to develop anthrax as a biological weapon. These weapons were specifically designed for cattle and were intended for use only if the Germans initiated a germ attack. Finely ground linseed meal cakes, hollowed out and sealed with powdered anthrax-causative agent, were created with the intention of feeding them to the cattle. By 1943, this project had produced over 5 million cakes, but luckily, they were never deployed. According to bioweapon projects' dominance, the former Soviet Union was at the top, dedicating and funding the development of biological weapons with a specific focus on anthrax more than any other nation. 3 facilities, Sverdlovsk, Penza, and Kurgan, were purposely set aside for this operation. Each facility was equipped with a 50-ton reactor. However, Sverdlovsk was the only active facility, while the other 2 served as mobilization capacities for potential increased production. The annual anthrax production estimate reached 5,000 tons by the 1980s. In March 1979, an accidental release of anthrax bacterium spores occurred at the Sverdlovsk facility, resulting in a death toll that remains unknown to this day. It is also worth noting that several countries, including Iraq and South Africa, pursued the development of biological weapons, particularly anthrax, albeit on a smaller scale compared to the efforts of the Soviet Union and the United States. In the 1980s, Iraq, led by Saddam Hussein's son-in-law Kamal, actively developed, produced, and stockpiled anthrax. This was evidenced by records from the American Type Culture Collection, confirming Iraq's purchase of several Bacillus anthracis strains in the late 1980s, before a ban imposed by the United States in early 1989. South Africa reportedly developed an anthrax weapon, as indicated by Dr. Schalk van Rensburg, a member of the Roodeplaat Research Laboratory. Dr. van Rensburg testified that South African security services attempted to poison 3 Russian advisors to the African National Congress with anthrax. Besides nations, individual terrorists and groups, such as the Japanese cult Aum Shinrikyo, utilized anthrax for various purposes. Aum Shinrikyo began working on an anthrax weapon in the mid-1980s, releasing anthrax on 3 occasions with no resulting human deaths. Their 1993 release in Kameido, Japan, was deemed unsuccessful as it used an attenuated or weakened strain of anthrax, Stern 34F2, meant for animal prophylaxis. It would be unfair not to mention the notorious US bioterrorism incident in 2001. On September 18th, letters containing anthrax pathogen were sent from the Trenton area to prominent figures, including NBC News anchor Tom Brokaw and an editor at the New York Post. The letters traversed postal facilities in Hamilton, New Jersey, and Morgan, New York City. Another 2 letters were then dispatched on October 9th, addressed to Senators Tom Daschle and Patrick Leahy, following the same postal route to Washington, D.C. Shockingly, a possible 3rd letter was intended for the American Media Incorporation, AMI offices in Florida, but was never located. This orchestrated attack resulted in 22 anthrax infections, exhibiting in 2 distinct clusters. The first from late September to early October, and the second from mid to late October. The initial cluster comprised 9 cases of cutaneous anthrax resulting from the September 18th letters, while the subsequent cluster involved 7 cases of inhalational anthrax and 1 case of cutaneous anthrax linked to the October 9th letters. Additionally, 5 cases, 4 inhalational and 1 cutaneous, occurred without direct links to the recovered letters, resulting in the tragic loss of 5 lives. An AMI employee in Florida, 2 Brentwood Postal Facility employees, an elderly woman in Connecticut, and a hospital employee in New York. This atrocious attack incurred substantial costs for the United States, reaching millions of dollars for containment, treatment, decontamination, and related expenses. The FBI, CIA, CDC, U.S. Postal Service, and Environmental Protection Agency were among the federal organizations that worked together to respond to the attack and carry out the subsequent investigation. This occurrence served as a crucial trial run that provided insights and lessons for handling a major potential bioterrorist incident. Sadly, the culprits behind this heinous act still remain unidentified. Plague is another potential biological weapon. It is a bacterial disease caused by the bacterium Yersinia pestis, with fleas and rodents serving as its reservoir. It is also a zoonotic disease, and its mode of infection is commonly through flea bites and aerosols. This particular disease has been linked to 3 historical pandemics: the Justinian pandemic, the Black Death or Great Pestilence, and the southwestern China modern pandemic. These 3 pandemics witnessed over 200 million deaths and adverse social and economic losses. All cases in these pandemics were in the bubonic form, originating from Yersinia pestis-infected rats and fleas, and also included the contagious pneumonic plague. The plague pathogen bears the traits of a bioweapon since it is highly infective and can be cultured easily in large quantities, increasing the likelihood of its later use. When dispersed, it also exhibits relative resistance to adverse environmental conditions and desiccation. The infectious dose estimate of this pathogen is between 100 to 500 cells. In humans, plague can manifest in diverse forms. The bubonic plague induces swelling and inflammation of lymph nodes, while septic plague triggers life-threatening systemic inflammation. The pneumonic plague is the most rapidly developing and fatal form. Pregnancy-associated plague can lead to fetal infection, and pediatric plague is prevalent in children. In earlier centuries, plague was employed as a biological weapon. For instance, during the 14th and 18th century wars, there were several cases in which plague-infested dead bodies were catapulted into enemies' camps. Additionally, the Japanese military, using low-flying planes, sprayed Yersinia pestis-infested fleas on Chinese civilians, leading to a massive outbreak of bubonic plague. This also triggered a vicious cycle of infection, as rats played a crucial role in its spread. By the 1960s, both the Soviet Union, USSR, and the US had active programs that could weaponize the plague-causing agent. However, by 1975, an accord under the International Biological Weapons and Toxins Convention prohibiting the use of bioweapons went into effect. Despite this agreement, the Soviets took their game to another level by developing Yersinia pestis that was resistant to antimicrobial agents in both dry and liquid forms. These strains were designed to be released via bomblets. Engineered to resist available treatment therapies, they posed a significant threat, which could have caused widespread infection and numerous deaths. Luckily, the Soviet research program on biological weapons came to an end around 1990. Another potential bioweapon is the bacterium Francisella tularensis, which causes tularemia, a zoonotic infection. The reservoirs of this bacterium are rodents, arthropods, and birds, and the main modes of its infection occur through tick bites, airborne transmission, or contact with its natural reservoir. The infectious dose estimate of this pathogen is between 10 to 50 cells. Despite being one of the most infectious agents, natural cases of tularemia in humans are quite uncommon. As early as 1932, the Japanese conducted research with this particular pathogen using prisoners of war. By the mid-20th century, both the Soviet Union and the United States had weaponized this pathogen, including its antimicrobial resistant strains. Numerous studies on humans and animals regarding tularemia took place at the U.S. Army Research Institute of Infectious Diseases at Fort Detrick, Maryland. After the 1972 Convention on the Prohibition of the Development, Production, and Stockpiling of Bioweapons, the United States destroyed its stock. Subsequently, the United States dedicated itself to defensive research programs, focusing on immunology, genomics, and vaccine production. According to the U.S. Centers for Disease Control and Prevention, the tularemia pathogen is likely to be used as a weapon due to its ease of dissemination, ability to cause disease, and immense negative impact on public health. However, tularemia is less likely to be preferred as a weapon compared to its counterparts due to the lack of a stable form, such as a stable spore phase. This makes it difficult to handle during production, with a high risk of infecting those involved in its production. In the event of a bioterrorist attack, the tularemia pathogen is likely to be released in the form of aerosols in a crowded area. Inhaling these aerosols can lead to pneumonic disease, oropharyngeal disease, and other complications. Besides bacteria, certain viruses have the potential to be exploited as bioweapons. Examples of this include the smallpox and hemorrhagic fever viruses. Smallpox is caused by the virus, variola major, with humans as its only natural host. Despite a lack of historical evidence showing smallpox as an effective bioweapon, its virus possesses traits that make it a potential candidate for terrorist use. It can be easily grown, protected from heat through lyophilization, and its genetic material can be modified as needed due to its large size. Moreover, it has the potential to be aerosolized. Possession of smallpox virus is illegal, and only 2 laboratories legally possess it: the CDC in Atlanta and the State Research Center for Virology and Biotechnology in the Novosibirsk region of Russia. The smallpox virus primarily spreads through respiratory secretions, resulting in an acute disease with a debilitating prodrome. It is one of the disfiguring diseases. The mortality rate of smallpox is approximately correlated with the number of skin lesions and, consequently, the severity of the previous viremia. It is roughly estimated to be around 30%. In the event of a smallpox outbreak, vaccination can be used as a preventive measure. However, there is no specific cure for the ailment. Years ago, President Bush ordered a large number of American healthcare and public health workers to undergo vaccinations in stages due to the possibility of a smallpox attack. Following the events of September 11, 2001, the US replenished its vaccine and vaccinia immune globulin (VIG) supplies, increased the number of laboratories equipped to test for the variola virus, and launched a comprehensive education campaign to better inform the public and medical professionals about the illness. Hemorrhagic fever viruses also pose a potential attraction for bioweapons. Briefly, these viruses are typically associated with fever and bleeding diathesis, and they normally belong to 4 families: Filoviridae, Bunyaviridae, Arenaviridae, and Flaviviridae. Dissemination of these viruses can occur through food, water, infected vectors, or aerosols. Reservoirs for some of these viruses include humans, rodents, mosquitoes, birds, bats, and primate ticks. Historically, these viruses have been weaponized by the United States, the former Soviet Union, and possibly by North Korea. The Ebola virus, Marburg virus, Lassa virus, Machupo or Bolivian hemorrhagic fever virus, Junin or Argentinian hemorrhagic fever virus, yellow fever virus, and Rift Valley fever virus were among the most researched and exploited viruses for this purpose. As we conclude, these are just a few pathogens that can be utilized as weapons of mass destruction, and the list can go on and on. For the sake of this video, let's pause here. Please let me know in the comments section if you would like a comprehensive video on viruses utilized as bioweapons. Thank you for watching.